Yogah Karmasu Kaushalam.
Hello friends, a very good day to you. I am Dr. Noopur Goyal from MG Science Institute, Ahmedabad. The topic of my today's discussion is bioremediation. Bioremediation is part of environmental microbiology, which in turn is part of environmental science. So, the first question is, what is bioremediation, or how do we carry out a bioremediation? This discussion is going to clarify certain points. If we talk about the historical perspective of this bioremediation, then the sewage treatment perhaps is the oldest type of the bioremediation. Even into the Egyptian civilization, Indus Valley civilization before Christ, we have the evidences of human waste treatment. This approach was into the early 19th and the 20th century was applied to the industrial waste. Before the 20th century, actually it was believed that solution to pollution is dilution, and hence the entire industrial waste and even to some extent human waste was discarded into the oceans, thinking that the vastness of the ocean is going to dilute and hence the pollution is not going to matter. But the mitigation of pollutants broke this myth, and we had to have some methods where the treatment had to be done. So around 1950s, this approach was of the waste treatment, human waste treatment, was extended to the industrial waste. Just a decade later, the need was imperative to find out the synthetic waste, because by that time the use of plastics, the use of pesticides became very rampant, and hence the waste generation because of all this novel compounds or the synthetic compounds also become rampant. In case of the 20th century and the late 20th century and the early 21st century, we started seeing the development into the bioremediation field very fast, and this resulted into the genomic approaches and other newer techniques which were being applied. After this history, let us see what is bioremediation. Bioremediation means application of biology to solve any kind of a problem. The word bioremediation is divided into two. Bio means biology and remediation. The meaning of the remediation is finding out a method to solve a problem, and hence bioremediation can be referred as process of using microorganisms or macroorganisms like earthworms, plants, etc. to remove the environmental pollutants, the toxic waste found into the soil, water and air. Here, why do we use the microorganisms specifically? The microorganisms are used specifically because they are in the food web, they come as decomposers or scavengers, and the versatility of the microorganism in terms of their enzymatic ability, in terms of the physical conditions into which they can grow is very vast. Microorganisms can be virtually literally applied in all the conditions depending upon the type of microorganism we choose. Say for example, we can find out a microorganism which is highly thermophilic, that is it can grow around 95 degrees C. We can find out equally the microorganisms which are anaerobic, that can grow without oxygen or under highly salinitic conditions, that are the aerophiles, or then we have the barophiles which grows under the high pressure, that is even into the bottom of the oceans they can grow. Because of this ability of high versatility as far as their metabolic conditions are concerned and their physiological conditions are concerned, microorganisms becomes the choice of organism for bioremediation. We will see into this further discussion what are the different techniques which are applied for the bioremediation, but let us see certain terms. Xenobiotic. Now xenobiotics are the organic compounds, but they are not found naturally into the environment. That is, they are not made into the environment. Similar term is the recalcitrant term, but the recalcitrant terms is a term where which can be biodegraded, but it is the biodegradation of such compounds, they require a very long time. They are very resistant to biodegradation, that are the recalcitrant, while the xenobiotic are foreign compounds. They may be biodegradable, they may not be biodegradable, but they are not naturally made. The another term which is of very importance is the biomagnification. Now this biomagnification is a process which would result into the imperative need for the bioremediation. Now biomagnification, it is a increase of a toxic compound, particularly a lipophilic toxic compound like hydrocarbons, metals, etc. Into the living tissue. Now what happens is, okay, the organism can ingest this lipophilic compound, they bind with the fatty layers into that, and they are never excreted out, and hence they keep on accumulating into the body. When into the food chain these organisms move upper side, then this enters into the another level of the food web. Say for example, into the ocean, the concentration of DDT, it is in the range of 0.2 ppb, but this DDT can be absorbed by the smaller plants, which are then taken up by the smaller fish. Smaller fish are eaten by the larger fish are being taken up by the birds, or the larger fish end up as an food for human beings, and thereby by the time this larger fish, it enters into the human chain or the highest consumer, the concentration becomes around 2 to 20 ppm, which is highly toxic. So this is what we mean by biomagnification, and biomagnification is the reason why we need to have bioremediation or biodegradation. Mineralization, it is a complete degradation of any toxic substance or any substance for that matter to carbon dioxide and hydrogen or water. The other inorganic compounds can be having a partial degradation, and intermediate stages would be there, but that phenomenon is also known as mineralization. The another term in reference with the mineralization is assimilation, which is exactly opposite of mineralization, whereby carbon dioxide and water molecules, they are assimilated into the higher compounds. Now, what are the principles of bioremediation? Bioremediation, basically it is based on to an idea that microorganisms, being so versatile, can utilize this compound as nutrients and they can grow on those compounds. But that is not the primary aim of the microorganisms. Suppose if it is an hydrocarbon, the way a hydrocarbon would be toxic to human beings, it would be similarly toxic to microorganisms also. So the first aim of the microorganism is to alleviate this toxicity. So they would try to degrade this compound. The best example we can give this is of HgCl2. Now HgCl2 is highly toxic to the microorganisms as well as the human beings. What microorganism does, this inorganic HgCl2, it has been converted into methyl mercury or methyl ethyl mercury. So they would no longer be toxic to the microorganisms themselves, but they can be toxic to the human beings. But mainly what we do is we try to find out the microorganism which produces the compounds which degrades the toxic substances and produces the compounds which are harmless in nature. The degraded molecule should not be harmful to the other life, and hence that has been there. Bacteria, protozoa, fungi are very well known for the transformation of product as a part of their metabolism that is being done. Bioremediation, it is a triple corner process whereby we can say that microorganism, plants and their enzymes play a very important role in degradation of both inorganic and organic pollutants which may be into the solid, liquid or gaseous phase. They are present into all different types of environments including soil, water and air, and bioremediation of all this can be possible. We can apply either the microorganisms which will be termed as for the degradation, biosorption or any other phenomenon. The process of bioremediation includes the release of enzymes to break down the contaminants into the digestible molecules. This contaminant of the organic substances are the ingest and digest as the food along with the energy source by the cell. The contaminant can be a source of carbon, but at the same time we must remember that they are going to require the source of nitrogen, phosphorus and other substances which can act as nutrients. What is our goal as far as the bioremediation is concerned? The bioremediation goal includes the conversion of hazardous waste to the inert products, or conversion of hazardous waste into the harmless biological substances which are no longer hazardous or toxic to the life. This is just a picture where it has been depicted that this is a microbial cell whereby the enzymes are there. The enzymes are secreted outside the microbial cell. The organic and other food contaminants are being taken up by the microbial cells, and final waste product are carbon dioxide and water. So this is just to give an idea of how a microbial cell can degrade the bacteria. I'm sorry, degrade the organic substances. Now, methods of bioremediation. Now, methods mainly can be divided into two broad categories. In situ bioremediation. In situ bioremediation is the remediation is carried out at the site of contamination only. That is, if it is a soil or it is an ocean, wherever we apply our technology, we go to that site, we optimize the conditions of the site, and we apply our microorganism into the site itself. While ex situ bioremediation includes excavating the site, bringing the soil, water or any other compounds to the laboratory or to the processing plant, and thereby carrying out the phenomenon of bioremediation. In situ bioremediation are of two types, that is the natural attenuation, that is we leave the site as it is, we don't do anything. The natural microorganisms which are present into the site would take care of the thing, and it would bring or restore the site which has been there. Now this is a perfect example which we had seen during our corona also. Everything was closed down, there was no vehicle and all, everything were being present. And hence we could find out that the quality of air improved significantly. That is, the air itself or the natural conditions itself took care of everything, and they were restored back to their original condition. So natural attenuation is something like that only. You leave them undisturbed and they will take care of it. But because our organic pollutants are so stubborn, it would take a lot of time, and in certain conditions we are not able to afford that kind of a time, and hence in situ bioremediation also we need to engineer the situation, we need to manipulate the situation. This manipulation of the situation can be brought about by biosparging, bioaugmentation, biological reactive barriers can be created, bioslurping can be done, phytoremediation can be done. Ex situ bioremediation is always engineered. We remove or we excavate the site, we bring the things, and then we apply either composting, land farming, or we have this constructed wetlands where only the bioremediation phenomenon is going on. Let us see what happens into the in situ bioremediation. Now this is, we have already talked about intrinsic means left on its own and it is done. Engineered in situ bioremediation, it is kind of a speeding up of the process. That is, the bioremediation process is speed up. The methods involved into the in situ bioremediation, it includes biosparging. Now, sometimes what happens is, see, we all know as far as the aerobic and anaerobic degradation is concerned, aerobic degradation is much more faster, much more efficient, and it is smellless also. The products which are being generated are cleaner products, and hence in all the conditions we do want aerobic degradation only, but the conditions may be such that the aerobic degradation is not possible. So because the environment is devoid of oxygen, or the amount of oxygen which is there, which is very less, and there is a huge competition into the amongst the microflora for that existing oxygen, and hence this biosporging, it is being done. All other conditions, they are what we call it as optimum but for the oxygen. So this is the power savaging we do. What we do is we inject the air under pressure and increase the dissolved oxygen into the groundwater. That is when we are interested in the bioremediation of groundwater, or we can apply this technique to the soil also. Now this is something like this, it is being done, and contamination, water table, it has been there. So because of the availability of the oxygen, rapid degradation of contaminants can take place. Now, biostimulation and bioventing are kind of similar terms which are being there. here, apart from supplying with the oxygen, what we are doing is we are adding the nutrients also. As we had said earlier, that one source of nutrient is available. Let's say, for example, we are talking about the hydrocarbons. So hydrocarbons can be a very good source of carbon, but what about the source of nitrogen, phosphorus, sulfur? That has to be supplied, otherwise the growth of microorganism would be inhibited. If there is no growth, there would not be any biodegradation, and hence we supply with them the oxygen and nutrients also. Biostimulation is a term we use when we provide this thing into the aqueous form and we allow them to circulate evenly into the entire area. Then we call it as a biostimulation. Bioventing is slightly different. We supply them with the air and nutrients, but what we do is into the contaminated soil, which is highly compacted, we first create the wells or trenches, and into these wells and trenches we add our nutrients and the oxygen, dissolved oxygen, it is there, and this can move forward. So that is being done as far as this indigenous bacteria are concerned. Then we have this bar augmentation. Now, the phenomenon of bar augmentation is slightly different. What we do here is the original source of microorganism is the indigenous bacteria or indigenous flora only. But what we do is we bring them into the lab, we try to isolate them separately, and we find out which is the main organism which has been doing, and we develop a consortia so as to increase the efficacy of this mixed culture, whereby they can act rapidly. So bioaugmentation is addition of developed microorganisms into the site with a hope that they will speed up the process. Along with the microorganism, addition of nutrients becomes must. Now, advantages of in situ bioremediation, they are extremely cost effective. Minimal exposure to public or site would take place. Then also the sites of bioremediation remains minimally disrupted. Disadvantages compared to ex situ, they are time consuming processes, and they are not that controlled also, because when we bring them into the processing plant, we can have a better control, while this in situ remediation would be exposed to the or would be affected by the environmental factors like temperature, oxygen requirement, pH, etc. But most effective is the or most affecting is the temperature, seasonal variations which are being there. So microbial degrading ability would also vary along with the season. So these are the disadvantages of in situ bioremediation, but as compared to ex situ, it is quite cheaper and hence more applicable into the situations. Now, ex situ bioremediation, now this is, it includes that what we do is we first identify the site which is to be bioremediated, and whether it is soil or whether it is the water, we remove this contaminated thing and we bring it to the processing plant, and thereby into the processing plant we have these huge bioreactors are set up, and into the bioreactors we mix the nutrients, we mix the microorganisms which are being there. The ex situ bioremediation can give us a better or efficient use of genetically manipulated organisms also. As I said, advantages includes better and controlled efficient processes. The process can be improved by enrichment with desired microorganisms. Time required is very less. It also allows the use of GMOs and etc. Disadvantages, the most barring process, or we can say the most hindering disadvantage is the cost. It requires a huge cost, particularly for the transportation and other things, which will be very cost, which will not be the cost effective things. Then the sites at which we are there, because of the excavation going on continuously, the sites are highly disturbed. And what about once the bioremediating is been done, then disposal problems are always huge. While in situ disposal problems do not arise because the site is there itself. What are the factors we need to take into the consideration which affect the bioremediation? The factors mainly are divided as biotic factors and abiotic factors. The biotic factors are the one which are the of the which includes the microorganisms. Now the microorganisms are the one which is going to play the important role, and hence the composition of microorganism in terms of what type of microorganism are present, what is the range of the microorganisms which are present, what is the amount of microorganisms which are being present, that matters a lot. Well, abiotic factors, there can be either physical or chemical. The physical characteristics mainly are the permeability of soil, porosity, etc., the presence of oxygen, pH, humidity, temperature, etc. The chemical factors is mainly the structure of the pollutant. Structure of the pollutant plays a very important role as far as the abiotic factor are concerned. The simpler and the easier molecule can be degraded faster as compared to the complex. If we talk about the hydrocarbons, then aliphatic hydrocarbons which are into the linear molecule can be degraded faster as compared to the aromatic molecules or the branched aliphatic molecules. Also, the presence of double bonds, alkenes and alkynes, the presence of double bonds and triple bonds would affect the biodegradable phenomenon. As the molecule becomes complex, its availability for degradation decreases. That is the main thing which has been there. The amount of salinity, because it is going to directly impact the growth of microorganisms, etc. The availability of the other nutrients like the source of nitrogen, source of phosphorus, sulfur, etc. also will affect the process. So these are been that. The temperature, pH, etc. would be affecting particularly the in situ bioremediation. Now, optimal performance of the biotic factors are directly dependent upon the abiotic factors. We may say that the biotic factors are into their optimum condition. Everything is there, but if the abiotic conditions are not optimum, then even the compound which is supposed to be degraded would become a recalcitrant despite the microorganisms being present. So abiotic factors play a very important role. I would say the abiotic factor plays an equally important role into the degradation factors. Now, types of reaction, they includes the aerobic reaction. Aerobic reaction are the one where oxygen becomes a final electron acceptor and the degradative molecules are carbon dioxide and water molecules. And this is the best type of the degradation which occurs into the shorter time. It doesn't produce any products which are of smelly nature, or any chances of toxic products are not being there. The toxic products would not be generated because they are the end products of this degradation is carbon dioxide and water molecules. Anaerobic bioremediation occurs in absence of microorganisms, in absence of oxygen, and they do not require the oxygen supply. So in under certain conditions like the depth of the soil or into the bottom of the lakes, this anaerobic bioremediation is been there. The most efficient anaerobic bioremediation we have seen in terms of biogas production, that is the methane generation. There are certain techniques which employs the anaerobic things like land filling, anaerobic sludge digestion. These all are the techniques which purposefully uses the anaerobic bioremediation. In case of anaerobic bioremediation, the molecules like nitrates, nitrites, sulfur, sulfide, they act as a final electron acceptor, and the chances are that we would have the end products as amines which are extremely smelly. The reactions which occurs are the hydrogenation, dehalogenation, carboxylations, etc. This is a figure which shows us the aerobic and anaerobic degradation of the same compound, that is the benzene as well as the toluene. The oxygenate has been there, the biomass is increased in terms of the number of bacteria, carbon dioxide and hydrogen are being produced. While here nitrate acts as a final electron acceptor, the carbon dioxide is produced, but at the same time nitrogen is also being generated. The water molecules are produced. Anaerobic biodegradation are seldom complete biodegradation. They are always the partially degraded molecules would be there and they need the further treatment. Now, Let us take the examples of certain bioremediation process. In that, the first one is the hydrocarbons. Now, when we talk about the bioremediation of hydrocarbons, we do talk about the bioremediation of both the pesticides which we are making and adding into the agricultural soil, and we are also talking about the oil spills, the crude oil which gets leaked, the refined oil which gets leaked. Simplest example we can give is all must have gone to the petrol pumps, and while they are filling the petrol into our vehicles, petrol or for that matter diesel into our vehicle, some drops of the petrol or diesel are always fallen down onto the what we call it soil, and that soil gets contaminated with the hydrocarbons. So hydrocarbon bioremediation of hydrocarbons deal with the soil which is contaminated with the petroproducts, and it also deals with the soil where excessive use of pesticide it is being done. Okay. We have given here the example of the Cold War between Iran and Iraq and America, where gallons of the petroleum was leaked into the sea, which resulted into the fish mortality. So oil spills can take place into the sea environment, which disturbs greatly the sea flora and fauna, and hence it has to be treated. Apart from that, the oil keeps on floating onto the sea, and it would reach to the beaches, or then they would result into the decreased value of aestheticism, and that then in turn tourism, and hence the economic loss would be huge, and hence the oil spills need to be treated. The main organisms involved into this treatment are Pseudomonas, Corynebacterium, Mycobacteria, and some yeast. The bioremediation of oil spills or the pesticides can be carried out by the developed consortia also, and by the use of genetically engineered microbial strains also. Uh, Here also the degradation process is affected by the composition of the oil spill. What type of aliphatic and aromatic compounds are present into the crude oil? That would be the major factor when we are using either the consortia or the genetically modified organisms for that. The structure of oil becomes very much important over here. Uh, Aromatic compounds are the ring compounds, and hence the little more recalcitrant as compared to the aliphatic molecules, and hence they would require, the first thing which would happen is the ring cleavage. As we can see, the first oxygenase reaction, it has been there, then dihydrobenzene is produced. To the dihydrobenzene we produce a catechol. Now from the catechol there would be a ring cleavage, and ring cleavage can be either at the ortho position or at the meta position. Once the ring cleavage is done, cis meconic acid is being produced, and this cis meconic acid would be further converted into organic acid, to aldehyde, to alcohol, which all the products can be finally subjected to the TCA cycle, that is tricarboxylic acid cycle. The end product of tricarboxylic acid cycles are always carbon dioxide and water molecules, and hence the breakdown of aromatic compound, complete breakdown of aromatic compound would result into the harmless products. Anaerobic biodegradation from the ring cleavage do not take place, but the substitution would be there, and we would have the benzaldehyde. Benzaldehyde would be then converted into the acetate or acetic acid, which would be then utilized by the methanogen bacteria to produce methane and carbon dioxide. This mixture of the gas can be used as a source of energy, that is in terms of biogas. Uh, Now, the genetically modified organism, the first it has been known as superbug, and the first superbug was created by Anand Chakraborty, Dr. Anand Chakraborty, and this was highlighted a lot into the thing, because into the 1980s when this was been created, to obtain the patent, because since it was a living organism, patent was not being granted automatically, and Dr. Anand Chakraborty had to move the American court for the same, and Diamond versus Chakraborty case was very highlighted at that time, and in fact, Chakrabarty was granted the patent of its strain, and this strain was very successfully used into the clearing of the oil spill which occurred into Valdez, Mexico City. Now what he did was, okay, he did this original strain he took was the Pseudomonas putida, and this Pseudomonas putida already contained the plasmid. Now plasmids are extra chromosomal substances which are being present into the bacteria on its own. So this contained a plasmid which had the ability to degrade the camphor. This organism was conjugated with a plasmid which could degrade the octane, and so we had one strain which had the ability to degrade the camphor and octane. The another bacteria was taken up where which had the ability to degrade xylene, and it was conjugated to with the bacteria which had the ability to degrade naphthalene. And the two bacteria we had now in totality, they could degrade all the four compounds. So the conjugation was done with these two organisms, and finally we had a superbug which had a plasmid for camphor, octane, naphthalene and xylene. So xylene represents the aromatic compounds, naphthalene represents the alicyclic compounds, octane represented the long chain aliphatic compounds, and camphor represent the complex compounds. And so between them they could degrade the entire crude oil structure. So this is how the superbug was being created, and it was widely used. But the problem with the superbug is this genetically engineered organism was found to be incapable of surviving into the natural environment. It could not survive, and always the risks of transferring these genes to the other organisms was always there. Secondly, it had to be supplemented with the oxygen, it had to be supplemented with the other nutrients which were being there, and so it became a little bit complex process. So instead, if we could develop the consortia, which was been a better story. So success with the consortia which was developed by TERI, Tata Energy Research Institute, New Delhi. Now they were termed as oil zappers. Two of their products, which were both working into the both the mesophilic range as well as the thermophilic range, that is at a high temperature, are extremely popular across the world. Not only in India, but across the world they are very popular. They are the oil zappers and oil zappers S. They are the consortia which have been developed from the site itself, and for the first time this technique was applied into the Mumbai offshore line in August 2010. Since then, as they say, the rest is history. After the success of oil zappers in 2010, we have not seen it back, but the oil zappers have worked wonderfully into the various countries as well as various things. We have taken one such example. Now this was a contaminated soil nearby the oil excavating wells in Mehsana. This is the pre-treatment thing. These soils were rendered, the fields were rendered completely useless, and this was the pre-treated soil. I'm sorry, I cannot go back to the slide, but I'm the other picture which was been there. The other picture showed us the treated soil. The next is the bioremediation of water. Now we have got two types of water. One is the surface water, that is the wastewater treatment. Now, as I said earlier into my presentation, wastewater treatment, that is the sewage treatment, is probably the oldest technology which has been applied very successfully in almost all the cities and everything. So here the collection of sewage, it is being signed, the sewage, it is being first filtered off into the Greek chamber, whereby the soluble and insoluble particles are being removed. Then we have this primary sedimentation tank where the soluble solids to some extent, and we mix it with the alum or another settling chemicals, so the entire solid matter would settle out, and the affluent, the solid matter, would be taken to the anaerobic sludge digestion, and the affluent would be given to the primary treatment. In the primary treatment, it would include the treatments like activated sludge treatment or trickling type filter type of the treatment which has been there. From there it would go to the settling tank, where the biomass which has been generated because of the treatment, that biomass would settle down, and that biomass would be taken up to the anaerobic sludge digester, and the affluent, it would be taken to the secondary treatment. Secondary treatment would include the removal of any kind of toxic substances, removal of excess of phosphate, removal of nitrates, making up of the total dissolved oxygen, which has to be somewhere around 7 milligram per liter. for it to be there, and then it would be chlorinated to remove any kind of or ozonated to remove any kind of the microorganisms which are live, and then it can be discharged into the nearby water body, or then it can be directly used as irrigation, cleaning purposes, etc. The anaerobic sludge, where the solid material would be digested, and as a result we would have a slurry which can be used as biofertilizers, and we would have a sludge which again can be used as a soil conditioner. In case of groundwater cleanup, both in situ and ex situ remediation would be there. In case of in situ remediation, what we will do, the polluted groundwater, we will add the nutrients, oxygen and the acclimated bacteria, that is the bacteria which we have developed into the laboratory, and we will hope that they would clear up the pollutants which are being present. But in case of ex situ, what we will do, we will remove this polluted groundwater, we will bring them to the bioreactor, and then we will clean up the entire thing. This is a tank which shows us the microorganisms and everything, and hence it can come into this thing. So from this bioreactor, we can add it into the groundwater, or we can remove the groundwater and we can bring it to the bioreactor. So that can be the technique which can be applied for the groundwater cleanup. Most of the time for the groundwater cleanup, the in situ technique is much more feasible into that. The chances of microbial contamination are always there, but the microorganisms which we are using are pretty harmless, and hence they can be used easily, or they can be used without giving much thought to them. Bioremediation of industrial waste have the similar process as the bioremediation of human waste, which has been there. Textile waste or any other kind of heavy metals, phenols, dyes, they are being used. This kind of bioremediation of industrial waste are highly specific processes, and hence they would require highly specific microorganisms. Most of the processes are patented also, and hence much detail is not available. But basically all the biodegradation, biosorption, leaching of the materials to bring them into the liquid form, all the techniques are being applied for the industrial waste. Our main aim over here into the treatment of industrial waste is to obtain some kind of a byproduct, so as to we can justify the effluent treatment, and we know the effluent treatment doesn't prove to be a drain onto the economic of the product. So either we can go for the generation of the biogas, that is the methane gas, either we can produce we can degrade the compound and we can produce a very good compost which can be sold either as fertilizers or as what we call it as soil conditioners. Then we can have this what we call it as electricitogens, that is the microorganisms which actually generate the uh, electricity. This is mainly used for the bioremediation of e-waste. Our discussion would be incomplete if we don't talk about the bioremediation of plastic. We do know that plastic is a huge nuisance into this today's era, which has been there. So two techniques are applied for the bioremediation of plastics. Biodegradation using the endophytes. Now see, basically what happens is endophytes are those molecules, those microorganisms which are which lives in symbiosis with the plants, and they are present within the plant cells, and hence they would be utilizing the molecules which are produced by the plants and all. The plastics have the similar chemical structure if we compare them with certain plant molecules, and hence the endophytes are already acclimatized to degrade such compounds. Endophytic fungus, like say for example, endophytic fungus, that is the Staphylococcus microspore, can degrade the plastic, both polyester as well as the polyurethane. The bioremediation of plastics would take a long time. We are talking in terms of years, 10 to 15 years are being there, which are being required for that. So the challenge, the biggest challenge over here is to speed up the process which has to be finished off within a month or so. So we need to find out more efficient organism for the same. Biodegradation by microorganisms, that is particularly the bacteria, can be there, which has been there, and they can utilize the plastics. Various strains of Pseudomonas and Bacillus have been applied successfully for the plastic degradation at the experimental structures. They found to be effectively degrading both polyethylene as well as polyethylene tetracycline PET. Both the plastic molecules are degraded equally efficiently by the help of this. Basically, here what happens is the Pseudomonas particularly, it colonizes onto the polyethylene and thereby start using it as a nutrient. Now, the another technique which is, now this is my personal favorite and hence I am using it. Now, microbially enhanced oil recovery, it is not exactly a bioremediation technique, not exactly a biodegradation technique, but it is a bioremediation technique. Now, this is a technique which is applied to the shutdown wells. We have recovered the oil from the wells, and now with the help of the conventional methods, we are no longer able to recover the oil from the wells. But this oil do contain more than 50% of the oil. So microbially enhanced oil recovery, we apply the microorganisms either for the direct oil recovery, that is directly the pressure would increase, or we prepare the well in such a manner that now we can apply the conventional technology. So basically what happens during the oil recovery is the types of oil recovery. It includes the primary recovery, whereby when the first time the oil is being discovered, the pressure into the oil wells are significant enough to mobilize the oil, and the oil automatically gushes out without the need to introduce any other substances into the well, and hence that is called as the primary recovery. It is a scaffolding type of a recovery where automatic oil is being recovered. The secondary recovery, it is oil is flooded with water or any other substances to increase the pressure or to flush out the oil which can be trapped or which is not easily recovered. So additional 15 to 20% of the oil would be recovered by this technique. Then the oil either becomes very porous, or the well would be the porous, or the well would become plugged and water molecule cannot work, because some one or the another reason we are not able to apply either the secondary treatment into the thing, because whatever the we do, the oil is not coming up. Under such circumstances, we can have a tertiary oil recovery, which is also known as microbially enhanced oil recovery. The use of microorganisms to recover at least minimum 10 to 20% of the oil can be recovered by this technique. Now, this technique also, what do we do in this technique? The microorganisms are introduced into the oil wells, and they are supposed to produce the harmless products such as natural substances, gases, which would push the oil out of the well, or which would act which would prepare the oil in such a manner that we can apply the secondary treatment or the secondary recovery to the well. What do the MUR do? Stimulation of oil production. It can stimulate the oil production directly. It can help into the wellbore cleaning so that we can apply with the secondary treatment. It can help with the selective plugging, where the channeling of the water would be done, and the water pressure which should be applied to the oil bearing rocks will not be there. This kind of the channeling can be stopped with the help of selective plugging. The production of biosurfactants, the production of microbial products like organic acids and all, they would dissolve the rocks and thereby release the oil which would be present into that, and that would help into direct oil recovery. The main microorganisms which are used applied are Clostridium. Now the Clostridium is one microorganism which is very helpful because it is anaerobic in nature, which is being used. Apart from Clostridium, other organisms which are being very successfully applied, it is what we call it is Xanthomonas campestris, that is the EPS producing organism which has been there. Talking about the techniques, stimulation of oil production, it carries out It is carried out by the methane and carbon dioxide production. This would result into the reduction of oil viscosity and increase of reservoir permeability. It would widen the fissures and the channels through the etching of the carbonaceous rock. This would allow the secondary oil treatment to the well, and the oil can be recovered. Wellbore cleaning, it is region by increasing the, or we will treat the well by increasing the average pore size. Various microbial metabolites like organic acids would be there. The organic acids would dissolve the various minerals like say calcium and the phosphate which are present into the rock, loosen the clay and deposits of the clay, and thereby release the oil which is trapped within the rock. The solvents, it would dissolve the precipitate organic deposit. Solvents would solubilize the crude oil also, and thereby release it into from and increase the oil pressure. Biogas, it would push the oil from the dead spaces where the water molecules cannot reach and the oil cannot be pushed out. Selective plugging. Selective plugging is done where the oil, well has become highly porous. That is, when we apply the water, instead of applying, increasing the pressure, the water gets channelized through that pores, and it would move out and become useless. So here we inject the slime forming microorganisms, that is the uh, microorganism that has the ability to produce a huge amount of extracellular polysaccharides which have been there. The most commonly used organism for this purpose is Xanthomonas campestris. If you don't want to use the Xanthomonas campestris, the product of Xanthomonas campestris, xanthan gum, will give us the similar result. What they basically do, they would go and bind, or they would select those sites which is extremely porous. They would grow there and kind of plug that site, and hence the equal pressure with the help of water can be applied. Now this is the what we call it as an uh, picture or the flow sheet of the well which is being done. These are the injecting wells where the microorganisms can be injected along with the nutrients and sometimes air, but air is seldom injected, but nutrients along with the microorganisms are there. When we are injecting the microorganisms themselves, we have to take into the consideration the environmental conditions which are present into the well, which can be highly anaerobic, highly saline. So we need to have the bacteria which are halophilic anaerobic organisms which will be there. Desulfovibrio, though a very useful bacteria in creating all, in satisfying all these conditions, unfortunately desulfovibrio produces a huge amount of hydrogen sulfide, which in turn is highly corrosive in nature and hence finds the limited use in case of MUR. Biosurfactants are the molecules which would decrease the surface tension of the oil, and hence the movement of the oil becomes easier, and hence they can be removed from the wells. MUR methods are of two different types. One, ex situ, whereby we grow the microorganisms into the bioreactor. We harness the metabolite like organic acids, xanthan gum, the gases which are being produced by them, and then we inject this microbial metabolites inside the well to carry out the MUR. In situ production is the injection of the microorganism itself into this thing. Ex situ production results into, or direct injection of the products resulting into the rapid MUR, while in situ production it would take a longer time to uh, carry out the oil recovery. Uh, which products are responsible for what? So biomass. Biomass are mainly used for selective plugging and reduction of oil viscosity. Biogas, that is the carbon dioxide, hydrogen and methane, it would increase the pressure inside the oil, and hence high energy potential would be created into the oil field and thereby help into the recovery of the oil. Hydrogen would increase the oil volume and decrease the oil viscosity, giving the better mobilization to the oil. Methane gas change the pressures into the potential fissures and pores, and hence the mobility of the oil would be increased and thereby increasing the production of the oil. Organic acids mainly which are being used are acetic acid and propionic acid. They can decrease the pH and increase the solubility of the rocks. It can also dissolve the carbonate rocks, and thereby the new flow channels can be created for the oil. Organic solvents, they decrease the interfacial tensions, and hence the fermented molasses into the pore canals, giving more oils from the rock. Bar lipid, they alter the rock feasibility, permeability, and thereby increases the wettability, break the oil water barriers, and improves the flow rate of the oil. Advantages of MUR, it includes the increased productivity of the oil field. The MUR setup, it is less expensive, and hence lower energy inputs are being required. It is most important part, it is environmental friendly, and microbial products which are being generated, they are biodegradable. And most of the time, see what happens is we have got nothing to lose. With a small amount of investment, we can recover the oil from the wells which are no longer working, and hence this has got an huge advantage for this. Disadvantages of MUR, it includes the we don't have basically the standardized framework which can be applied for the evaluation of microbial activity, which microorganism is giving what. It is still into the trial and error method, and secondly, this technology is highly site specific. The technology which works for one well may not be similar, so it has to be optimized for each and every well individually. The exogenous microbes, it requires facilities for the cultivations, the entire setup of bioreactors and everything would be required. Uh, Oxygen, if at all we are interested in the aerobic or microbially enhanced oil recovery, then oxygen itself would act as a corrosive agent, and thereby economic loss in terms of non-resistant equipments and pipelines would take place. Plugging of reservoirs due to aggregation of the cell mass or the precipitation of metabolites is also known. And besides, the groundwater pollution is always a thing. Now this study was been made in Poland, whereby when it was been found out that the MUR was applied to one well, and because of the migration of the microorganism, after 6 months or a year, it had been observed that oil pressure is increasing into the neighboring wells also. This was a result of the migration of microorganisms. So this again very useful as far as oil recovery is concerned, but it also leads to a question mark of groundwater pollution which is being there. Despite all this, there is a MOU signed between ONGC and TERI, where TERI has developed this MUOR, which has been oil O, given name as oil O, and it has been successfully applied onto the, initially it was applied to the 82 wells which are located into the Mehsana district, and then after the success of this, it has been now, it was applied to the 50 wells which have been located into the Assam. So ONGC is very easily using this kind of an technology for this kind of things. So this, which why we have taken up this example of MUR. See, basically the shut off oil wells as such are not causing any pollution, but the bioremediation technique, if they have been applied to this, we can with the minimal of investment, we can gain a product, or we can increase the productivity of the oils. And we all know how we are in how much crude oil is essential for us, and we can save a lot into the foreign exchange money if we can become self-sufficient into the crude oil production. So if we can apply this technology, if we can standardize this technology for all the oil wells across the country, then probably we may become self-sufficient. The other environmental issues which occurs because of this is the subsurface water, that is the migration of organisms of or metabolites into the groundwater. The huge amount of waste water which has been which is created because of this, washing of the equipment, spent fluids, media and chemicals used for the pH adjustment, they all would generate the waste water, and the effluent treatment would require the capital investment. then hydrogen sulfide can be produced, and this would lead to the environment pollution also. But despite all this, developing methods for the MUR, they are being growing, and the bacteria are improving, lowering the production cost, genetical modification can be done, the enzymatic ability of the bacteria can be enhanced using several other techniques, and hence they can be proving better and better alternative, or we can say supplementary to the chemical methods. The another technique, which is bioleaching or biomining. Bioleaching or biomining, it is the use of naturally occurring bacteria to extract metals from their ores. Basically here, with the help of microorganisms, the insoluble metal sulfides are converted into water soluble metal sulfates, which are then recovered easily. Bioleaching was started in late 1940s, and then it has become a worldwide phenomenon. Bioleaching is mainly used to recover copper, zinc, lead, arsenic, uranium, nickel, molybdenum, gold, silver, cobalt, etc. And it is very widely used in, of course, it is used in India also, but other countries like US, Australia, Canada, Zambia, Indonesia, application of bioleaching is far wider. The microorganisms used, mainly the bacteria, the chemolithotropic bacteria, which is Acidithiobacillus peroxidans and Acidithiobacillus thioxidans, are the main organisms which are being applied for the bioleaching. Newer techniques of the bioleaching do make use of several fungi like Aspergillus, Trichoderma, etc. Uh, uh, all different types of bacteria, they are being used. Then extreme thermophiles includes sulfolabus bacteria. The mechanism of bioleaching, what does it do? The bacteria oxidize the ferrous ion and sulfur to produce ferric ions and sulfate. While it is producing the sulfur or the ferric ion, they in turn reacts with the sulfide minerals to produce the metal sulfate, which is soluble, and ferrous and sulfur. This is a list of various organisms which are being applied into the different pH range as well as the different temperature range into the mining. As you have seen, it mainly includes Bacillus peroxidans, Lactospirillum peroxidans, that is the iron and sulfur bacteria play a very important role into the bioleaching. Techniques of bioleaching again are in situ leaching, that is into the mine itself it has been done, and ex situ leaching. See, up till now bioleaching has been applied onto the abandoned mines, whereby no longer the pyrohydrometallurgical techniques are economically feasible to extract the metals. Metals are there, but extraction of metals by the conventional method is no longer economically feasible or economically viable, and hence these techniques are applied, or these techniques are applied to the mines where the metals are there but they are of very poor quality, and hence conventional methods prove to be costlier. So these techniques are applied to this thing. Ex-situ, it includes heap or dump leaching or the bioreactor leaching. Both techniques are used equally well. In situ leaching, it has been applied to the mines where they are impervious to the water. That is, the surface of the mine has to be non-porous, or it should discourage the penetration of water into that. Now here basically what we are doing is we are inserting two pipes, perforated pipes. As you can see, this is the perforated pipe which is carrying the microorganisms for the leaching. It is carrying the low pH nutrients for the leaching which has been there. These organisms, they would spread out into the mine, they would carry out the leaching, and the leachate which would be generated, which would contain the soluble metal, that would be onto the slightly onto the lower side, and another perforated pipe, this would bring to the carried the leachate to the recovery substances. Here the metal would be recovered. The leftover leachate which would be there, which would be made up with the total what we call it as whatever the substances which are being lacking, the source of nitrogen, the ferrous, which would be added, mixed. It would be mixed with the microorganism and it would be added, reused again. So this is the in situ mine leaching. In situ mine leaching, it is a very good technique, but it takes a lot of time for the bioremedy, biomining thing. But sometimes what happens is, if the abandoned mines are left as it is, and if they are particularly open mine, then this kind of an harnessed bioleaching becomes important, because otherwise they would lead to the problems like acid mine drainage, which in turn would lead to the acid rains. Now, in case of dump leaching, what we are doing in case of dump leaching, the ore are excavated from the mine, asphalt pad, a layer of plastic it has been there onto the thing, and the perforated pipes are laid over the asphalt pad with the connections onto the dump. Huge dumps in terms of millions of tons of ores are being created, and the trenches are created into this dump. Air ventilation system are also provided, because these organisms are there, and we have this the methods, recycling tank, where the microorganisms, nutrients, sometimes acid, it is being added so as to pH can be adjusted, and they are being sprayed over the ore, and they are allowed to act. As they would act, they would solubilize the metals, and the liquor which would be containing the soluble matter, they would be collected into a lake-like body which has been created. And from this we call it as an pregnant leach solution, which means that it contains a huge amount of metals, and then they go to the metal recovery plant, where the metal would be recovered, and whatever the effluent which would be there, that would come into the recycling of the solution. And just like in case of mines, here also we make up the solutions, we add the required microorganism, required nutrients, and we allow it to recycle. So this, compared to in situ leaching, this process is faster, which can be done. But as again, ex situ leaching, it is costlier also. This is an bioreactor leaching, more controlled process which has been there, where we can control each and every parameter which has been there. Here the metal sulfide low grade ores are brought to the processing plant. They are first crushed so as to increase the surface area. They are added into the reactor. From the reactor, continuous reactors can be there, and leachate would be collected, and this leachate would be then given to the extraction. The extraction methods includes electrowinning, solvent extraction, biosorption, bioprecipitation, etc. And then metal would be extracted, that would be sold, and whatever the effluent which is left out, they can be reused. Advantages: as all other bioremediation technique, bioleaching, it is also a simpler, cheaper method to operate and maintain. The process is more environmental friendly and than the traditional extraction method. Uh, it can reduce the amount of greenhouse gases. It can be used for the mines which are uh, too poor, or which have the quality of the metals which are too poor to the technology sense. Bioleaching is applied to the for uranium leaching. Now uranium leaching is very much profitable, in the sense that it would act as both the purpose. It would recover the uranium, which again is a highly precious metal, and also it would decrease the toxicity because of the uranium. So radioactivity of the uranium can be decreased with the help of bioleaching. Disadvantages: unlike all the other bioremediation processes, it is extremely slow process. The heat created because of the growth of microorganisms can kill the bacteria. Toxic chemicals sometimes are produced. Secondly, all the microorganisms which we are using for the bioleaching purpose, they are extremely fragile, and hence the metal, as they are getting into the dissolved concentration, the concentration of metal increases, the activity of microorganism can decrease. The examples of bioleaching which are being there in India, they are been established. Of course, Hindustan Copper Limited, they are applying this into their zinc mines also, and copper mines also. Then the bioleaching plants have been established in Hyderabad and Bangalore also. Jharkhand has a bioleaching plant for uranium. In Gujarat, we have the Ambamata mines, which are under the with the GMDC, and since the copper quality is not that much proper, the bioleaching it is being done, and then the gold mines in Karnataka, bioleaching is being applied. A lot has to be done in case of bar leaching. We need to find out the new bacterial strains which would survive into the extreme environments and hence give a better bar leaching processes. We need to find out the microorganisms which can tolerate or which can grow despite the presence of higher concentration of minerals. Then we need to find out the organisms, or up till now we are doing the biology with the help of acidophilic organisms only. We need to find out the bacteria, or we need to create an environment where a neutral or slightly less acidic environment is been there, so that we can apply other organisms also. And of course, use of genetically modified organisms is always there. Uh, The other, apart from the microorganisms, these are few cases which we have discussed, but apart from the other microorganisms, the plants can be a very good source of bioremediation. We have these indoor plants, where we can say if they are there, they will purify your air. That is nothing but a type of phytoremediation. The bioremediation which is carried out by the help of plant, it is called as phytoremediation. Now, phytoremediation, it includes mitigation of pollutant from the contaminated soil, water and air. Plants mainly work as absorbent of these nutrients, and then these plants can be disturbed, what we call it as disposed of. They would act as an biosorbent. The pollutant would get accumulated into the plant itself. Uh, so for example, these are the plants. Two things can be happening. The plant cells can degrade the pollutants directly, which is true in cases of simpler pollutants. But most of the time, the plant roots, they would absorb the pollutants and they would be present into the plant. The plant can be taken up, it can be burnt off as a biomass, or any such technique can be applied. In case of metal removal, the plant can be used to recover the metals also. Up till now, we have identified more than 400 plant species which would naturally take up the toxic materials. For example, a sunflower plant was been very successfully used in the Chernobyl site, where to remove the radioactive cesium and strontium. Water hyacinth have been used to remove the arsenic in India as well as Bangladesh, where arsenic is a huge problem into the drinking water. Genetically modified plants have been used to remove the trinitrotoluene and RDX from the war sites also. The drawbacks of phytoremediation, it is as usual a slow process. The plant has to first grow, and then it would be taking part into the remediation. Secondly, with the help of the plant, only the surface soil can be decontaminated. We cannot use the plant for the subsurface soil or the deeper soils. Mycoremediation is application of fungi for the remediation. The primary role of fungi is the decomposition. Fungi has the inbuilt ability to degrade the complex molecules like cellulose, lignin and pectin, and hence they can be used to remove the complex organic and inorganic molecules also. The key to microbioremediation is choose a right fungal species to target a specific pollutant, and hence it also requires a lot of pre-study before it can be applied. Certain fungal strains have been reported successfully degrade the gases like VX and sarin, which has a complicated effect on or toxic effect on our nerves. More fungi are more widely used as biosorbents to remove metals from effluents. Fungi are also used for the degradation of dyes, particularly into the textile plants. They are being very successfully applied. Microfiltration, it is a kind of an filter. It has been created with the help of fungal mycelia, and these filters are then placed into the soil as well as the water, so as to absorb the chemical polluants. Now, this whatever are the current thing, but what are the recent trends in bioremediation, how they are being applied? So enzymatic bioremediation. See, we know the growth of microorganism, it is slightly a complicated process. So what happens is the microorganism are highly sensitive to the seasonal variation of the things, the temperature, the pH and everything. And the growth of the microorganism would take a longer time. Once they'll grow, they will secrete the enzyme and then they will carry out the degradation. Here what we are doing is, instead of growing the microorganisms and then by creating the problems of ecological issues and other thing, we are directly using the enzymes, a wide variety of enzymes like oxygenases, lactases, peroxidases, dehalogenases, esterases, phosphotriesterases, lipases, cellulases, xylanases can be applied directly into the pollutant environment, and this polluted environment would be treated with the help of enzymes. This would reduce our time, and it would also take care of the problems of horizontal gene transfer among the bacteria and all the other things. Enzyme technology have been perfected, and hence low cost enzyme production techniques are already mastered, so we can use this technique for the bioremediation also. Gene editing, now with the help of CRISPR-Cas9 or with the help of ZNFs, TALEN, there are certain techniques which have been there, whereby what we are doing is we edit our gene so as to produce the better enzymes, so as to increase the efficacy of the bioremediation process. The microbe produced as a result of gene editing now would work into a shorter time period, it would produce more enzymes, and it would be kind of resistant to the toxicity of the pollutant, and hence it can act upon the more toxic product also. So gene editing tool can be utilized very efficiently for creating a potent microbe which would be able to degrade the pollutant into a shorter time. Genetically modified organisms are always being used, but see, this genetically modified organism, though it is a recent trend to use them, we should always be careful regarding the two things. One is the conjugation phenomenon, which is very common in bacteria, and horizontal gene transfer to the non-target organism would always be a risk. And second thing is the survival of GMOs into a pollutant environment. Okay, so that has been that. But transgenic microorganisms, transgenic plants are being applied for the remediation purpose. Uh, as I said, it is a double-edged sword and hence has to be used very carefully. Nanobioremediation. This is an amalgam of bioremediation and nanotechnology. Here, with the help of the nanotechnology, we are creating the nanomicroorganisms which can be then fitted into the several different shapes like nanotubes, nanosheets, and then they can be applied for the bioremediation. This use of nanobioremediation, it is one technology which can be used with the same efficacy in situ remediation as well as the ex situ remediation of the harmful components. Then apart from the initial costing, the application is less costlier, and hence and it is speedier also, hence they can be used for the thing. Overall advantages of the bioremediation. See, in terms of advantages, we can say bioremediation is the only technique available to us into today's era, because we need to go green, we need to save our earth, and hence the bioremediation is the technique we need to apply. So no additional disposable cost, low maintenance, did not create any kind of an matlab can be applied directly onto the beaches or anything. The aesthetic value of the site does not decrease, capable of impacting source zone and thus decreasing site cleanup time, and hence keeps the site disruption to the minimum. Very important in case of tourist spots. Disadvantages of the bioremediation is, first and the foremost, is the time consumption. It is extremely slow process. So we need to have the technique which would speed up this process. The other disadvantages is it is not an universal technique. It is always a site specific technique, and for a particular site the bioremediation technology has to be optimized. So which has been there. All the hazardous waste cannot be treated. Some waste like metallic waste are too toxic for the microorganisms, and microorganisms do not grow in their presence, and hence they can never be treated with the help of bioremediation. We have several barriers which are there, like say for example research barriers. The microorganism can be very potent in laboratory, but when it comes to its application into the field, it doesn't give that much promised, it doesn't give whatever it has promised into the lab. Technical barriers, same, the technology transfer is not that efficient. In lab we are very efficient, but when we apply it to the field, it is not efficient. Economic barriers, some of the technology they are very good but very costly, and hence we are not ready to apply it onto the wastewater systems. And of course government regulatory barriers are always there. Uh, Concluding my thing, I would only say that bioremediation is a need of time, and hence we need to apply the bioremediation wherever possible. We need to go green, we need to have this biofertilizers, biopesticides, and thereby we can have this, we can save the earth. As far as the bioremediation is concerned, such a future prospectus, mainly it includes the development of non-culturable bacteria. See, soil is considered to be the storehouse bacteria, and hardly 1% of the soil bacteria can be cultured. So what we need is, we need to find out the techniques apart from metagenomics where we can identify the role of, exact role of the soil bacteria into the bioremediation, and thereby develop efficient consortia to mitigate this problem. One of the things which we can do for development of the efficacy of bioremediation process is identifying the MCPs, that is the methyl accepting chemotaxis proteins among the bacteria. If we can identify this for each and every pollutant, then the bacteria would migrate towards the chemical pollutant automatically and thereby degrade it efficiently. Uh, we need to develop more efficient enzymes also for the utilization, because enzymes in lieu of plants and microorganisms can prove to be a very efficient, very potent, and less time consuming solution to bioremediation. These are several references which I have used, and thank you for your patience listening. And thank you for your patience listening. Have a nice day.